that's my fault, sorry.
Mm.
But Petromyzontida here is the lamprey. Within the lamprey, again, there's a specimen here. There's also a specimen over there in that box. The lamprey, again, has 4 parts you need to identify. It's the same 4 parts that we just got through discussing with the lancelets. Again, muscular postanal tail, pharyngeal slits, the notochord, and the notochord. Simple enough?
Mm-hmm.
We got it? All right, keep moving. Here we go. The next clade is chondrichthyes. Chondrichthyes are the cartilaginous fish. Within the chondrichthyes, we've got the rays and we've got the sharks. That's really all we're going to ask here. So just make sure that you can identify these as chondrichthyes, this as a chondrichthyes, this as a chondrichthyes as well. Got it?
Awesome.
Good. All right. Next group. Within the bony fish, we have Actinopterygii. Actinopterygii is the only clade within the bony fish we have. We don't have any lobe-fin fish, right? And we don't have any lung fish. Lung fish, again, are in Dipnoi. Lobe-fin fish are the Actinista. We don't have either one of those because, again, they're very, very rare. In fact, they don't even occur within the waters around North America. So all we have is Actinopterygii. So everything here in this particular container, Actinopterygii. Still going. Next class, right? I'll go ahead and call this a class. Up to this point, I've been calling them clades. Here, most people recognize Amphibia as a class, so we'll just use the word class. Within the class Amphibia, we've got basically the frogs and the salamanders. So again, take a look, make sure you can identify that as an amphibian. You also have a salamander here. It doesn't look much like a salamander. It always confuses people whenever I put it on the exam. FYI, most likely will be on the exam. So take a look at it, make sure you can identify it as an amphibian. Got it?
Okay.
You're like, oh, that must be like a lancelet, because it kind of looks like that thing over there in that jar, except it has legs, at least in the front half. Okay.
So you just gonna ask us what class is it, and then we put okay.
Stop trying to make the video
Yes, you
longer,
remember the
man.
previous lab exam? I know it was a long time ago, but the previous lab exam where
I only have 45 minutes.
and we asked you basically what phylum, what phylum does this belong to, and the phylums were Mollusca and Porifera and Cnidaria and all those phyla. It'd be just similar here, right? What clade or class does the organism belong to? All right, the other thing with an amphibia, you have to be able to identify the parts of the bullfrog. So, there's several bold words there, one of which is not in the long list, the tympanum. What's the tympanum? It's that big eardrum thing there on the side of the head, okay? Your tympanic membrane is inside of your external pinnae, that means your ear. Your tympanic membrane, your tympanum, is internal. On the bullfrog, they don't have external ears, they have that membrane there that functions the exact same way. So again, that's the amphibians. Now, we move on to the reptilian/avian clade, right? Again, birds classified as reptiles now. Within the reptiles, it's phylum Chordata. I think we knew that though, right? Phylum Chordata. Yeah. Come on in. We care about you. Come on in. You're laughing because we said we didn't care about anybody who's not here. But we care about you because you're here now. All right. Now, what we have here within the reptiles, make sure you understand the reptile diversity, or at least the diversity here within the reptilian clade. Within the diversity of the reptilian clade, basically we divide all the reptiles into what we call the archosaurs and the lepidosaurs. What are the archosaurs? I'm just kind of setting you up here. What are the archosaurs? We have the turtles, we have the crocodilians, and we have the the birds, right? So the archosaur lineage within the reptiles, these cameras all
You're a celebrity.
It's okay.
Within the reptiles, we again are within the archosaur lineage. We have the turtles, right? Most likely, again, at least according to molecular evidence, we've got the turtles, we've got the crocodilians, and we've got the birds. Now, on the other half of the reptilian lineage, we have the lepidosaurs. What are the lepidosaurs? Squamates and
Sea squirts?
Snakes.
The squamates were the lizards and the snakes. Now, the other half of the lineage is the tuataraes, right? So again, that's the diversity that we find within the modern day extant species of reptiles. Extant implying that they're not extinct. So again, turtles, crocodilians, birds, and then you've got the squamates, the lizards and the snakes, right? We group those together, and then the tuataraes. So there's 5 groups of extant reptiles. There's a question on that. on your exam most likely. So that's why I'm kind of covering that now. Now, the only other thing here that you have within this particular grouping, right? Again, make sure you can identify those where they go within what we just discussed. These are all reptiles, right? So snakes. The only other thing here is, well,
Hi, Alex.
the only other thing here is we had that discussion on some act of legislation that basically protects birds.
Yes, we did.
What was that called?
The Migratory Bird Treaty Act.
The Migratory Bird Treaty Act, right? Do you remember what the Migratory Bird Treaty Act protects? It protects birds, obviously. It also protects their eggs. It protects their feathers, and then it protects their nest, right? So we have the Migratory Bird Treaty Act that protects birds, but it also protects bird products like the nest, the egg, and the feathers. Now, I don't know if Dr. Armenta's class went into it, but what are the exceptions to the Migratory Bird Treaty Act?
Non-native species.
Non-native species are not protected, right?
Hunting.
Hunted species are not necessarily classified. And then special permit birds can also be collected as well. Okay? So again, those are the exceptions there that would allow for somebody to actually go out and kill birds. Moving on, the class Mammalia. The class Mammalia. Now, within the class Mammalia, make sure you're familiar with the three clades of mammals. They're in bold. The first one is monotremes, right? So within the monotremes, we have the platypus and the echidna. Do we have any specimens of those?
No.
No, right? We don't have any of those. We do, however, have this particular clade. This clade is marsupial, right? Again, this is a opossum. Opossum is a marsupial. Everything else up here is a eutherian. Perfect. You got it?
Mm-hmm.
One other thing here, and then we'll be down to lab 10. The last thing within lab 10 is you've got two mammal dissections. We did not do the pig. We can ignore the pig. We're not going to look at the pig. You've got the rat dissection, so make sure you look over all the parts of the rat, right? All those parts there in bold in that list. And then we also have in there the cat dissection. Okay? Guys, again, frogs, rats, cats, lamprey, they're all out and available. So if you want to look at any of that stuff, you've got the next hour to do so. The cats are in the box over there. Everything else is over there in that little, I don't know, that big plastic tub. Got it? Okay, now, for the rest of it, for lab 11, okay, let me summarize what we got here for lab 11. For lab 11, the ecological studies lab, make sure you understand what behavioral ecology is, right? Addressing the evolutionary significance of why behavior exists. From that, we then moved on to the method of estimating population growth. Growth being kind of the key word there. So how do we actually go about estimating population growth? That's where we look at the raccoons, right? We start with 2 raccoons. They have 2 raccoons a year. They have 4 at the end of the year, but those 2 are not reproductively mature until their 3rd year. That's when they have young. You're going to have a scenario that is not identical to that, but somewhat similar to that, and you're going to have to estimate population growth. Now, in addition to that, why are those numbers not realistic to life? In other words, why is that not real? It doesn't include death. It doesn't include the idea of limited resources and carrying capacity. It's like I'm being burned by red lights. It doesn't include here the idea of limited resources, which establishes the carrying capacity, right? So understand what carrying capacity was. And then also the idea of immigration and emigration. So the movement into a population, movement out of a population is ignored in that particular scenario. So that could affect the overall numbers at the end of the equation. Moving from that point on, we then talked about estimating population size, right? So not necessarily the growth, but the actual size of the population. There were 2 approaches. One was called the density extrapolation. The other was called the Mark recapture technique. Density extrapolation is pretty simple. We take the density of a known area and we multiply that number by the total number of areas. Right? Like, oh, I don't know. Sometimes it works, but you know, we'll do the example here, right? Diana, how many people are at your table? 6. How many tables in the room? So what's our total Help her out, Daniel. 24. Okay, so somebody count the number of students in the room.
We can't count that.
Somebody say 26. 25?
25.
Good job. You estimated very, very well. So if there's actually 25 in here and you estimated that there were 24, could we accept an error of 1? So what did we just do there? She knew the density of her table, and she multiplied that by the number of tables, and that gave us an estimated population size. Make sense? That's what we call the density extrapolation method. Now, the second method is the mark recapture technique. Number 1, right before steps number 1, we go out and we capture animals. Number 2, we go out and we actually take those that we capture and we mark them, right? So we put a band on them or who knows what. Number 3, we release them, and then number 4, we go out and capture again. That gives us 3 numbers that we play with. Number 1 is basically the number of individuals that we caught the first night. Second number is the number of individuals we caught the second night. And then the third number is the number of individuals that we caught on both nights. Make sense? So if we caught 10 the first night and we caught 10 the second night, and between the 2 nights we had 2 recaptures, what's our estimated population size? Let's try that again. If we got 10 the first night and 10 the second night, but there were 2 that were caught on both nights, what's our estimated population size? 50. There you go, right? The formula for that is capture 1 times capture 2 divided by recapture. So 10 times 10 would give you 100, divided by 2 would give you 50. Got it? Cool. All right, wrapping up this lab now. We had the various traps. So real fast. What's this trap called? It's little, but what's this trap called?
Box trap.
Give me more specific. It's a Sherman, right? So Sherman box trap. What would this be used to catch? Small mammals, right? Rodents, things like that. What was this one called?
Freaky.
That's the tomahawk live trap. Tomahawk live traps, have hard traps, similar, same thing, more or less. Again, this particular trap used to catch medium-sized mammals, right? Again, these vary in their size. These can be small, these can be great big. In fact, I've seen ones large enough, just like this, one's large enough to catch wolves. Right? It's funny, you could actually get 3 or 4 people with those traps. It would be fun to set out here, you know, on the campus. Put like, you know, final exam in there. All right, this one, the Maccabee gopher trap, right? So again, we set the trap, put it down in the hole, and it catches the gopher. That's the only example of the kill trap that I actually have out here. Okay, a dip net, right? Dip net to dip something out of the water or to catch something on land. And the last one that I have out is, what's that called? The ball shot tree, right? So the ball shot tree trap here, again, is a trap that you would use live bait in. So you put live bait there inside of a little cage. There's the door that opens. Stick it in, close the door. The little mouse frantically runs around. The red-tailed hawk comes down and, oh, it caught him, right? Or that's magic, one of the 2.
Awesome.
So the little fishing line slip knot there on top of the bow shot tree trap catches the talons of the hawk, and he's now caught. We can do the same thing except without bait. We can wrap a log in the pond or in a river that turtles will crawl out onto, and turtle claws
Man, I need a
and
tripod,
legs will
my
actually
hand
be caught
hurts.
up in the exact same concept. So you can go along and just pull the turtles out. Bow shot tree. Got it? All right. That wraps up lab 11. Lab 12 is pretty simple. Ready for lab 12?
Mm-hmm.
Lab 12? Yes? Okay, here we go. That's lab 12. We'll make this like in 2 seconds. Make sure you understand what biodiversity is. Make sure you understand what species richness is, what relative abundance is, and then make sure that you know how to calculate Shannon diversity indices. Right? Make sure you can calculate the actual value there of the Shannon diversity indices. And that's it.
Awesome.
Anything,
Stop video.